हे फक्त हेच टेलिकास्ट होतं आहे ना, बाकी कॅमेरावर नाही. So good morning everybody. Today we will study the physiology of skeletal muscle. Now if you remember, we started this topic with the structure of a neuron. We studied various parts and functions of the neurons, the nerve fiber, and then we studied the neuromuscular junction. So once a neuron is stimulated and an action potential is initiated in the initial segment of the axon, and it is then carried along the nerve fiber to the axon terminal, and the axon terminal ultimately forms the neuromuscular junction. It releases a neurotransmitter which we call as acetylcholine, and then this acetylcholine brings about an excitation of skeletal muscle. So now before proceeding any further, let us study the process of the physiology of the skeletal muscle. These are our specific learning objectives. To know, number one, to know the types of muscles. Number two, the structure of skeletal muscle fiber. Number three, myofibril and arrangement of myofilaments in a myofibril. Number four, sarcomere and its bands. And finally, the detailed structure of myosin and actin filaments. Now the muscle tissue, as you know, it forms almost half the weight of the body mass, has the ability to change chemical energy into mechanical energy. It's something like a transducer. So here the chemical energy comes from breakdown of ATP, and the mechanical energy that is formed is either a shortening of the muscle or increase in the tension in the muscle. Perhaps you are all aware that there are three types of muscular tissues. They differ in structure, location, functions, and the means of activation. That is how they are activated. The three types are the skeletal muscle, cardiac muscle, and the smooth muscle. Skeletal, cardiac, and the smooth muscle. The skeletal muscle are very long, cylindrical, multinucleate cells with obvious striations. So as you can see, these are the cross striations. So the skeletal muscle is also called as striated muscle. And then we have the cardiac muscle present in the heart, which has a branching chains of cells and striations. And the smooth muscle, which is, these are the single muscles, spindle shaped, uninucleate. There is single nucleus and there are no striations. The skeletal muscle or the striated muscle attaches to the skeleton. Microscopically, it has stripes, and these stripes are called as striations. So as you can see, these are the cross striations across the muscle fibers. So that is why they are called as striated cells. And this is under voluntary control, that is under conscious control. These skeletal muscle cells are multinucleated. Cardiac muscle present in the heart also shows striations, but these striations have branching patterns. So as you can see over here, these are the branching patterns coming or present in the striated muscles. Naturally, this is involuntarily controlled, it is not under voluntary control. Usually there is one nucleus, but there may be more. And the neural control allows the heart to respond to changes by changing the rate of contraction, the force of contraction, etc. So whatever is the output of the heart, that can be altered by changing the heart rate and as well as the force of contraction. The smooth muscle are present in the viscera, in the blood vessels, tubular structures like the gastrointestinal tract, the urinary tract, also present in the eye, and so on. It helps substances move through internal body channels via the peristaltic type of actions. Also control the diameter of the blood vessels and hence blood pressure. So this is one of the important aspects of the smooth muscle. They control the diameter and therefore the peripheral resistance and therefore the blood pressure. They are not striated, they are not under voluntary control, and there is a single nucleus. Now let us proceed with the details of the structure of skeletal muscle. So you'll know about the muscle cell itself or the myocyte, the coverings of a muscle, the structure of myofibrils, arrangement of myofilaments in myofibrils, and the basic concept of sarcomere and its bands. So these are the various points about the skeletal muscle which we'll go into the details of. Right. The skeletal muscle fibers form 40% of the body weight, 10% about smooth muscles. The cardiac muscle and the smooth muscle together make up about 10%. So totally the muscular tissue forms about 50% of the body weight. The muscle consists of large number of individual cells, and each cell is called as a muscle fiber or a myocyte. This is because the muscle fiber is an elongated structure, elongated thread-like or fiber-like structure, hence it is called as muscle fiber. Each muscle fiber, please note we are talking of a single myocyte. Each muscle fiber is a long cylindrical cell with multiple nuclei just beneath the sarcolemma. You know the cell membrane of the muscle is called as sarcolemma. So there are multiple nuclei and overall shape it's a long cylindrical thread-like structure, that is a myocyte or a muscle cell. These muscle fibers are 10 to 100 micrometers in diameter. So the diameter of a skeletal muscle cell can range from 10 micrometers to 100 micrometers, and the length can go up to 30 centimeters. Each skeletal muscle cell is a syncytium produced by fusion of embryonic cells, that is the myoblasts, and that is why these cells are multinucleated cells. So what is present in the cytoplasm of the muscle cell? The cytoplasm contains, number one, large number of mitochondria. You know, the process of contraction as well as the process of relaxation is a active process, and you know any active process needs energy, and therefore there are large number of mitochondria present in the sarcoplasm, that is the cytoplasm of muscle cell. Number two, the endoplasmic reticulum. The endoplasmic reticulum in the skeletal muscle is modified to form the L tubules of the sarcoplasmic reticulum. Of course, sarcoplasmic reticulum means the endoplasmic reticulum in a muscle cell, and of this sarcoplasmic reticulum, most of the tubes are arranged. Basically, you know, endoplasmic reticulum is a tubulovascular structure. It is a network of tubulovascular structures. So here the tubules are arranged longitudinally, that is parallel to the length of the muscle fibers, and therefore these are called as the L tubules, L for longitudinal. So the longitudinal or the L tubules are the modified tubules of muscle endoplasmic reticulum. Number three, there are granules of glycogen present inside the cells. So the glycogen, you know, is a source of formation of ATP. Then there is an oxygen binding protein called myoglobin. This myoglobin has a P50 value which is very low, indicating that it has a strong affinity or strong capacity to bind oxygen even at low oxygen levels. Then there are ribosomes and lysosomes, and then the muscle cells contain myofibrils containing myofilaments. Myofilaments are the microfilaments. You must have heard about microtubules and microfilaments present inside the cell. So the myofibrils, which contain myofilaments, and of course the additional part is the T tubule or the transverse tubules, which are formed by invagination of sarcolemma. So at some places the sarcolemma dips inside or into the interior of the muscle cell, forming a tubule which runs across the length, and therefore these are called as the transverse tubules. So there are L tubules, that is the sarcoplasmic tubules, and there are T tubules or the transverse tubules, and together these two structures, they form what is called as sarcotubular system. They form sarcotubular system, and the sarcotubular system is essential for spread of excitation into the interior of muscle fiber. Each muscle cell is covered by a thin layer of fibrous tissue called as endomysium. So if you see here, this is the single muscle fiber as shown here, and the single muscle fiber is covered by a thin fibrous layer, and this thin fibrous layer is called as endomysium. So each muscle cell itself is covered by a thin layer of fibrous tissue called as endomysium. Many such muscle cells are grouped together or joined together to form muscle fascicles or muscle bundles, and these muscle bundles are covered by another layer of fibrous tissue which is called as perimysium, and large number of muscle fascicles are grouped together or they all join together to form a muscle which is covered by epimysium. So there are three fibrous layers covering the muscular tissue. One is the endomysium covering the individual cell, then there is a perimysium covering the fascicles, and there is epimysium covering the entire muscle. At either ends, the muscle fibers are attached to fibrous tissues in the tendons, which are attached to the bones. Right. So this is the single muscle fiber covered by endomysium. These are the muscle fascicles, as you can see, and these muscle fascicles grouped together, they are covered by perimysium, and then many such muscle fascicles coming together are covered by the fibrous tissue which we call as epimysium. So these are the coverings of the muscular tissue. Each muscle fiber contains many elongated cylindrical subunits called myofibrils. Now this is a cross section of a single skeletal muscle cell, and you can see nucleus here as well as here, and then you can see that inside at the transverse section you can see there are many cylindrical units, and these cylindrical units are called myofibrils, and they are about 1 micron in diameter. So the diameter of each myofibril is around 1 micrometer, and these myofibrils contain the contractile filaments of the skeletal muscle cells. The adjacent myofibrils are separated from each other by endoplasmic reticulum, that is the L tubules, and the organelles. So note that inside this myocyte, through the cross section we can see many of these cylindrical subunits. We can also see some mitochondria and the tubules. But note that there is no membrane covering a myofibril. There is no membrane covering this cylindrical structure myofibril. The adjacent myofibrils are separated by the intracellular structures like the L tubules, like the mitochondria, and so on. The myofibrils make up about 80% of the skeletal muscle volume. So obviously the remaining 20% is occupied by the cell organelles and the vesicles and mitochondria and so on. The arrangement of myofilaments in a myofibril creates a series of repeating alternate dark and light bands along the length of the myofibrils. So inside the myofibrils there is some arrangement of myofilaments or the microfilaments, and they are arranged in such a way that the cell appears to have cross striations, and that is why we call this a skeletal muscle. Right. Now, what are the different types of microfilaments? There are two types of microfilaments that are present inside a myofibril. One called thick filaments or myosin filaments, and two thin filaments or actin filaments. So each myofibril contains two types of microfilaments, thick or myosin filaments and thin or actin filaments. These filaments are arranged in a specific manner. Each myofibril is divided into a number of segments by certain transversely arranged disc-like fibrous structures called Z discs. Now look here, this is one of the Z discs. This is the next Z disc. So these Z discs are transversely arranged across the length of the muscle. The portion of the myofibril between two successive Z discs is called a sarcomere. I'll repeat, the portion of the myofibril between two successive Z lines or Z discs is called a sarcomere, which is the functional subunit of a muscle, and it has a length of 2 to 2.2 micrometers. I'll repeat, the portion of myofibril between two successive Z lines or Z discs is called sarcomere, and in the resting state the sarcomere has a length of 2 to 2.2 micrometers. Right. Now again, this is a diagrammatic representation of the myofilaments as well as the sarcomere. You can see that there is a Z disc. This is one Z disc at one point, this is another Z disc at the other point, and the portion of the myofibril between two adjacent sarcomeres is called as a sarcomere. So here also you can see this is the Z line or Z disc on this side, this is the Z disc on the immediate next Z disc, and the distance between the two successive Z discs is called as a sarcomere, and in the resting state this length is about 2 to 2.2 micrometers. The central portion of the sarcomere, before that, the myosin filaments are present in the center of the sarcomere, while the actin filaments or the thin filaments are situated at its periphery. So you can see here, this is one Z disc, this is another Z disc, so this is a sarcomere, and you can see that in the central portion of the sarcomere is the thick filament or the myosin filament shown with the red color here. Here. On the other hand, the thin filaments which are shown here with the blue color are the actin filaments or the thin filaments, and as you can see, these thin filaments are attached to the Z discs on either sides. So these are the actin filaments or thin filaments attached to this Z disc, these are the actin filaments attached to this Z disc, and the myosin filaments are floating in the central portion of the sarcomere. The peripheral ends of actin filaments are inserted into the Z disc and their central ends partially interdigitate with the myosin filaments. So here the peripheral ends are inserted into the Z disc, the actin filaments on this side into this Z disc, the actin filaments on this side into this Z disc, and you can see that they partially interdigitate into the thick filaments. This arrangement of filaments produces alternate light band and dark band along the length of myofibrils. The central portion of sarcomere containing myosin filaments and the central ends of the actin filaments is called dark band or A band. A refers to anisotropic, where the polarized light is refracted in more than one direction. So this is anisotropic band or A band. So this central portion containing the myosin filaments and the interdigitating portion of the actin filaments, all this appears dark under the microscope. So here also you can see this is the central portion from this point here to this point here. So this central portion of the sarcomere contains the thick filaments of the myosin filaments plus the interdigitating portion of thin filaments. So this band consisting of the myosin filaments as well as the interdigitating part of actin filaments appears dark under the microscope, and therefore this is called as the dark band or A band, A for anisotropic band. The central portion of the dark band containing only the myosin filaments appears lighter and is called as H band or H zone, which is seen in the relaxed state of the muscle. So here you can see that in the central portion there are only myosin filaments. There are no interdigitating actin filament. They end here itself and here itself. So the central portion of the myosin filament, there is no interdigitation. And because there is no interdigitation, it contains only the myosin filaments and therefore it appears lighter in color. So this central portion of the dark band, which appears light because there are no overlapping A bands, is now called as H band. And in the center of this H band there is a line which is called as M line, and this M line represents the connective tissue which is interconnecting the adjacent myosin filaments. So the central portion of the dark band containing only the myosin filaments appears lighter and is called H band or H zone. This is seen in the relaxed state of the muscle, so that the actin filaments are pulled out of the myosin filaments and this zone appears in the center. In the center of the H zone, there is a thin line called as M line which I showed you just now, and this is formed by the filamentous structures transversely connecting the central portions of myosin filaments. So these filamentous structures help to maintain the position of the myosin filaments. The portion of myofibril between two dark bands appears lighter, and hence it is called as a light band or isotropic band, that is I band. So the part of the myofibril between two adjacent dark bands which appears light because there are no myosin filaments in this, and therefore it is called as light band or isotropic band because polarized light comes out only in one direction. So this I band consists of peripheral portions of two adjacent sarcomeres containing only actin filaments and the Z disc in its center. Right. So here you can see that this is one dark band. This is the edge perhaps of the next dark band. So this portion, which appears lighter in color, as you can see over here, and which contains the Z disc in the center and the non-overlapping portions of the actin filaments which are inserted into the Z disc from either sides. So this is called as a light band. So the central portion of the sarcomere containing actin and myosin is called as dark band or anisotropic band, whereas the portion of the myofibril from between two adjacent dark bands appears light, and therefore it is called as I band or the isotropic band, and in the center of the isotropic band there is a Z disc, and you know that on either sides of the Z disc are these actin filaments which are inserted into the Z disc. So this particular arrangement of the thick and the thin filaments causes the appearance of these bands. The central dark band or A band, anisotropic band, contains the myosin filaments as well as overlapping portions of actin filaments. In the central portion of the dark band there is a H band or H zone where there is no overlapping between actin and myosin, and in the central portion of this A band again there are these filamentous structures connecting the myosin filaments and holding them some sort of scaffolding, and so this forms the M line. And the light band between two adjacent dark bands is called as the light band or the I band because it is isotropic to the polarized light. Naturally it will contain the Z disc in the center, and on either sides it will contain the actin filaments, some belonging to this sarcomere and some belonging to the next sarcomere. All the myofibrils in a muscle are arranged in such a way that their Z discs are in the same transverse plane and are attached to one another. So now imagine here, this is one myofibril with the Z disc, this is another myofibril with the Z disc, there is a third myofibril here with the Z disc. All these Z discs are in the same transverse plane. All the Z discs are in the same transverse plane and they are attached to each other, and therefore the dark bands of all the myofibrils in a muscle fiber, they form dark bands or dark striations across, and therefore the skeletal muscle is called as a striated muscle. So the alignment of adjacent myofibrils is such that the Z discs of the next adjacent is in the same plane as the Z disc of the next, and thus all these Z discs are in one line, and therefore all the dark bands are in one transverse line, all the light bands are in one transverse line, and therefore you see the cross striations in the skeletal muscle. Thus each sarcomere consists of one dark band in its center and two half light bands at periphery. You can imagine this. One sarcomere consists of one dark band in the center and two half I bands on either side. So together they make up one sarcomere. In addition, there are certain filaments called titin filaments which connect the ends of the myosin filaments to the Z disc and maintain alignment of myosin filaments amongst the actin filaments. So you can see certain filaments which are called as the titin filaments. You can see these titin filaments, they are attached to the peripheral ends of the myosin filaments, and at the other end they are attached to the Z discs. So these titin molecules are perhaps the largest molecules in the body, and the titin filaments ensure that the thick filaments are maintained in their position in the sarcomere. In addition, there are certain other proteins like Actin N the actin protein helps in attachment of the actin filaments to the Z discs. So the actin filaments, which are not shown over here, they help in attachment of actin filaments to the Z discs. Right. Now let us go a little more into the details of muscle contraction. We'll consider first the structure of thick filament or the myosin filament. Then we'll consider the structure of myosin molecules which form the myosin filament. Then we'll consider the structure of actin filament or the thin filament, the components like the F-actin, the tropomyosin and the troponin. And finally we'll study the orientation of proteins in actin filaments in presence of calcium ions and in absence of or under low concentration of calcium ions. Right. So we start with the discussion of about the structure of myosin filaments or the thick filaments. Each myosin filament is 1.6 micrometers long and has a diameter of 15 to 16 nanometers. I'll repeat. Each myosin filament is 1.6 micrometers long and has a diameter of 15 to 16 nanometers. So you can see over here, this is the thick filament. The length of this filament will be 1.6 micrometers and the thickness or the diameter of the filament is 15 to 16 nanometers. Because this is thicker as compared to the actin filaments, so these filaments are also called as myosin filaments or the thick filaments, their diameter being more. Now this myosin filament is formed by polymerization of about 200 myosin molecules. So myosin molecule, one of which is shown over here, this is the structure of a myosin molecule, and 200 such myosin molecules are organized or are arranged together or polymerized together to form myosin filament. I'll repeat, about 200 myosin molecules polymerized to form myosin filament. And the length of the myosin filament is 1.6 micrometers and the diameter is 15. So the length is 1.6 micrometers and the diameter is 15 to 16 nanometers. Each myosin 2 molecule, now whatever we are describing is myosin 2 because it has two heads. Anyway, we'll discuss that later on. Each myosin molecule has a molecular weight of 480,000. Each myosin molecule has a molecular weight of 480,000. This is the structure of a myosin molecule. It is a hexamer, that is, it has 6 units and made up of 6 polypeptide chains. So basically these are the chains of amino acids. So there are 6 polypeptide chains in a myosin molecule. There are 2 heavy chains, each with a molecular weight of 200,000, and there are 4 light chains, each with a molecular weight of 20,000. Now you can see here in this diagram, these are the 2 heavy chains or the long chains, as you can see, and each heavy chain has a molecular weight of about 200,000. There are 4 light chains, each with a molecular weight of 20,000. Of these 4 light chains, 2 are the regulatory chains which regulate the ATPase activity of the myosin head, and 2 essential chains or the alkali chains which stabilize myosin head. Right. The 2 heavy chains wrap spirally around each other to form a double helix. So here you can see that these are the 2 heavy chains which are extending right from this part right up to the head, and these two heavy chains are wrapped spirally to form the tail of the molecule. So this part, which contains the two heavy chains wrapped around each other, this portion is called the tail of the myosin molecule. One end of each of these heavy chains project outwards and is folded into a globular protein called myosin head. So you can see here, one end, see, this is the other end of the heavy chain, and this is the first end of the heavy chain. Here at its end, the chain is folded upon itself to form a globular structure. So this is the head of one thick chain or heavy chain, and this is the head of the other heavy chain. So this folding of the head forms a globular structure, and therefore it is called as head of the myosin molecule. Thus there are two heads in each myosin molecule. The other end of the coiled double helix is straight and is called the tail, which we have already discussed. Each myosin head also contains two light chains. So each of these myosin heads contains two light chains, that is, basically it is one end of the heavy chain plus there are two light chains in each head, so totally 4 light chains in one myosin molecule. These light chains help control the movements of the head during the process of muscle contraction. I'll repeat, these light chains which are present in the head, globular head portion of the myosin molecule, contains the light chains. Each head contains 2 light chains, so totally there will be 4 light chains of it during the process of muscle contraction. The projecting part of the heavy chains is called a cross bridge, consisting of an arm and a head. So this part of the myosin molecule, which projects outwards from the direction of the tail, away from the direction of tail, this projecting part is called as the cross bridge. It is called as cross bridge because during the process of contraction, the head of the myosin molecule attaches to the actin molecule, and therefore this is a one that interlinks these two parts, and therefore it is called as a cross bridge. And the cross bridge consists of an arm and a head. So this is the head, and this is the outward bending portion of the heavy molecule. This is the arm. So there is a head, there is an arm, and there is a tail. So these are the physical part, let us say, of the myosin molecule. Each cross bridge is flexible at 2 points called hinges. One where the arm leaves the tail, and the other where the head attaches to the arm. So you can see here in this diagram, the cross bridge has 2 hinges, one at the junction of the head with the arm, and the other at the junction of arm with the tail. So one flexible point at the junction of the arm with the tail, and another at the junction of head with the arm. So these points which are flexible, they are called as hinges, and these hinges allow the head of the myosin either to extend far outward from the body of the myosin filament or to be brought close to the body. So you can see from the diagram that these hinges permit the bending of the head portion towards the tail or bending of the head portion away from the tail. So these movements of head are essential for process of contraction as well as relaxation. So these are called as hinges, and they allow a relative movement between the head and the arm and the tail. These hinge points or hinge heads participate in actual contraction process. Now functionally, there are two binding sites on the head of myosin molecule. One for the attachment to the actin filament. Remember, the myosin heads have a strong affinity for actin filaments. And number two, it has a binding site for ATP, that is adenosine triphosphate. So, as far as the structure of the myosin molecule is concerned, it is a hexapeptide containing 2 heavy chains and 4 light chains. The light chains are also called as regulatory chains. The heavy chains at one point project outwards and they form the arm and the head. The arm and head together are called the cross bridge, whereas the remaining straight portion is called as the tail of the molecule. The head of the myosin molecule has 2 functional sites, one for attachment to the actin filament. Basically, because the contraction of muscle occurs by the attachment of myosin filament to the actin filament and then their interaction, and the other is for binding of ATP. So it is interesting to note that the head of the myosin itself has ATPase activity. So whenever ATP binds with the head of the myosin, the ATPase activity is automatically stimulated and the ATP is broken down and the released energy is used to bring about the movement of the filaments. So the head of the myosin has 2 binding sites for attachment, one for actin and another for ATP, and the head of the myosin also has ATPase activity itself. About 200 such myosin molecules are arranged in such a way that the tail portion of all the molecules are bundled together, forming the body of myosin filament, and the cross bridges project outward in all the directions. Let us see the diagram. Right. So you can see the arrangement of these myosin filaments. The tail portion, which is the straight portion of the myosin filament, it polymerizes together to form the body of the myosin filament. And the cross bridges or the heads, they project outwards from this body of the myosin filaments and they extend towards the direction of the thin filaments or the actin filaments. About 200 such myosin molecules, that is the whatever single molecule structure we have studied, 200 of such myosin molecules polymerize in such a way that the tail portion of all the molecules is bundled together forming the body and the cross bridges project outward in all the directions. In the body of myosin filament, half of the myosin molecules are arranged with their cross bridges extending in one direction and the other half of the molecules with their cross bridges in opposite direction. So as you can see over here, the central portion of the myosin filament does not contain any cross bridges. The reason for this is that it contains only the tail portions of the myosin molecules. It contains only the tail portions of the myosin molecules, and from remaining part of the myosin molecule on either sides project the cross bridges which interact with the actin filament. So note that in the central portion of the sarcomere or in the central portion of the myosin filament, there are no cross bridges because it contains only the tails of the myosin molecules. And then on either sides, the molecules are polymerized in such a way that the cross bridges of the half of these myosin molecules or myosin filaments project in this direction, half of them project in the opposite direction. But as you can see, if these myosin heads interact with the actin filament on this side, and if these myosin molecules interact with the actin filament on this side, when the heads of the myosin filaments, myosin molecules start flexing, you can imagine that this actin filament will be pulled towards the center of the myosin filament. These actin filaments will also be pulled towards the center of the myosin filament, and thus the actin filaments from the two sides will be brought towards the center of the myosin filaments, and that will cause the shortening of the muscle. So there are no cross bridges in the center of the myosin filament for a distance of 0.2 micrometers because the cross bridges extend towards both the ends of the myosin filament away from the center. Hence, in the center there are only tails and no heads of myosin molecules. Now during the process of muscle contraction, myosin filaments interact with the actin filaments and drag the actin filaments towards the center of the myosin filaments as we discussed just now. So the muscle achieves shortening by pulling the actin filaments on either sides towards the center of the myosin filament, and that is how the size of the sarcomere or the length of the sarcomere is going to reduce, and when the length of all the sarcomere reduces, it reduces the length of the muscle, that is the muscle actually contracts. Thus, actin filaments slide into the myosin filaments, leading to shortening or the contraction of muscles, and this is called as sliding filament mechanism of muscle contraction. So sliding filament mechanism says that during the process of muscle contraction, the cross bridges of the myosin filaments pull the actin filaments towards the center of the myosin filaments or towards the center of the sarcomere, and this sliding of the filaments leads to shortening of the muscle. Myosin heads have strong affinity for active sites of actin filaments. This point I have already mentioned earlier. Right. Now coming to thin filaments or the actin filaments. The actin filament has a length of 1 micrometer and a diameter of 5 to 6 nanometers. 1 micrometer, that means they are shorter than the thick filaments because thick filaments are 1.6 micrometers. And here they are narrower, and that is why they are called as thin filaments because their diameter is 5 to 6 nanometers, whereas the thick filaments have 15 to 16 nanometers. Now these actin filaments are composed of 3 different types of proteins. Number 1, protein called actin. Number 2, protein called tropomyosin. And number 3, a protein called troponin. So actin, tropomyosin and troponin together polymerize in a specific manner to form the actin filament or the thin filament. The ratio of these 3 proteins is like this: 7 actin molecules, 1 tropomyosin and 1 troponin. The actin part of the thin filament forms the backbone of actin filament. It is a double-stranded F-actin protein. The word F stands for filamentous or fibrous. So basically, there is a actin filament which is formed by the double strands of F-actin proteins. Each strand of F-actin helix is composed of polymerized G-actin molecules. G stands for globular, that is because it is a spherical in shape, and this globular G protein or G-actin, it is 3 to 4 nanometers in diameter, and each G-actin has a molecular weight of 42,000. I'll repeat. Now look at this particular diagram. You can see that there are 3 different types of proteins. Number 1, the blue colored structures are the actins, and you can see these actins, there are 2 strands of actins, and these strands of actins are formed by joining together of G-actin molecule. So this blue colored spherical thing is the G-actin molecule. This blue colored is the G-actin molecule, and like that. So these G-actin molecules join together to form F-actin filament. And there are 2 such F-actin filaments. You can see this is one, and you can see this is the other. And these 2 F-actin filaments, they twist around each other to form this base of the actin filament. I'll repeat. The actin filaments or the thin filaments are made up of three types of proteins: F-actin, tropomyosin and troponin. F-actin strand is formed by polymerization of G-actins or globular actins, and these G-actins joining together form the F-actin, and there are two such F-actin strands which twist around each other, forming the base of this thin filament. There are 13 G-actin molecules in each revolution of F-actin strand. So you can see here, this is the one end and this is the other end of the twist, as you can say, and therefore there are 13 G-actin molecules in each revolution of F-actin strand. So if you say that the revolution begins here and then it is completed here, there are 13 G-actin molecules in one revolution of F-actin strand. Totally there are 300 to 400 G-actins in each actin filament, and each revolution having 70 nanometer length. So this, from this point to this point, it is 70 nanometers in length, and in one revolution there are 13 G-actins polymerizing, joining together, forming two F-actin strands which are twisted around each other. Each G-actin molecule is having ADP, and these ADP molecules are believed to be the active sites or the binding sites of actin filaments with which the cross bridges of myosin filaments interact to cause muscle contraction. The second protein is tropomyosin. The tropomyosin molecule has a molecular weight of about 70,000. The length of the tropomyosin molecule itself is 40 nanometers, connected end to end to form a ribbon-like strand called tropomyosin strand. So here, if you look at the diagram, here the tropomyosin molecule is an elongated molecule. Each molecule has a molecular weight of 70,000 and a length of 40 nanometers, and such tropomyosin molecules join end to end to form a tropomyosin strand. These tropomyosin molecules join end to end to form a tropomyosin strand, and there are two such tropomyosin strands present on either sides of groove formed by the double helix of two F-actin strands and loosely connected to them. Right. So again, let us go through this. Tropomyosin molecule is an elongated thread-like molecule, molecular weight 70,000, length 40 nanometers. These molecules join end to end to form a ribbon-like strand which is called as tropomyosin strand. And there are two tropomyosin strands in each actin filament present on either sides of groove formed by the double helix of F-actin strands. So the G-actins join together to form F-actin. Two F-actin strands twist around one another to form this base of the filamentous actin, and when the two filamentous actins twist, they form grooves on either side, and therefore there are two such tropomyosin strands present on either sides of the groove formed by the double helix of F-actin strands and loosely connected to them. In the resting state, the tropomyosin molecule physically covers the active sites of actin filaments. Yes. Now you can see here the dark portion in each spherical G-actin filament is its active site. You can see these dark portions, and these active sites in the resting state, they are covered by these tropomyosin strands. and therefore tropomyosin, because the tropomyosin is covering the active sites of actin filament, the myosin heads cannot bind with the active sites of actin filament. So in the resting state, the tropomyosin molecules physically cover the active sites of the actin filaments so that the interaction cannot occur between actin and myosin filaments to cause any contraction. Each tropomyosin molecule covers 7 active sites. That completes the description of tropomyosin, and in the next class we will discuss the structure of the third protein present in the actin filament, that is tropomyosin. So thank you, and we will continue in the next class. Thank you. Yeah, sanitizer.